Good morning, folks. It's time for our everyone's favorite class, Neuro3301. Um, we love a dark morning, don't we, folks? We love it when you wake up and it's pitch black outside and freezing cold. We love it. It's great. It's wonderful. It's a great time of year. Everyone's mood, I'm sure, is at it's peak right now. Everyone's feeling like things are really under control and not at all starting to slip into seasonal patterns of negative affect. At least that's what I hope. Anyway, we're back. We're back at it. We're talking about Huntington's disease today. So the next two lectures, this one and the one that's going to come on Monday, they are all about trinucleotide repeat disorders. So we're taking a bit of a break, or we're making a bit of a diversion, or we're transitioning or something. We're changing over from epigenetics, which was the sort of focus of our talk on drug addiction, which mainly just the things that happen in your brain epigenetically when you're exposed to drugs. Now we're switching gears into some very classic, very, very, very genetic disorders where you don't even need to do a GWAS to figure out what's happening. So for the next two classes, we're going to be looking at things in that perspective. So, I guess hopefully just giving you a sense of the different ways that genetics and epigenetics can influence mental health or, neurological conditions. On the one hand, you can have things like schizophrenia, where a whole bunch of things act in a polygenic way to bring about or to increase your odds, say, of having that condition. All the way over to here, where the presence or absence of a given mutation guarantees you'll get the condition, and it can be genotyped very, very easily. So, just trying to give you a little bit of a flavor for all the different varieties of ways things can play out genetically. Let's get started. So here's our lectures, our title screen. We're talking about Huntington's disease. Sometimes it's also called Huntington disease. I'm not really sure what the modern day preference is. I always learned it as Huntington's disease because it was named after a guy named Huntington. But it's HD, you might want to call it. So as I was saying, many of the disorders we've talked about so far and that we will talk about are what you describe as polygenic, meaning that there's no simple singular gene you can point to that is responsible for you having or not having the condition. Rather, there are many, many different genes or many, many different locations in the genome that all have a relatively small individual effect, but when they're added together, they change, appreciably change your likelihood of having or not having a given condition. Huntington's disease is not like that. It is monogenic. It's one of the most common monogenic neurological disorders in the world, meaning that it's caused by a single gene. It affects about 1 in 7,300 people among the Western populations. It's difficult to sort of ascertain the prevalence worldwide, because you need to do genetic testing to confirm an HD diagnosis. Although I think you can look at the pedigree and the family tree and whatnot, and you'll see that it's pretty obvious just from the way it's inherited. But so like I said, most of the time, family history, you can just kind of look at the pattern of inheritance and just the way the pattern of its onset in patients, and it looks pretty much like Huntington's, although about 5 to 8% of cases might be de novo mutations. So the way Huntington's plays out in real life is it begins with a gradual decline in motor function that you would start to see around early middle age, and it will reach a fairly advanced state by the mid-50s. So the motor deficits consist of what's called chorea. This is, it actually used to be the disorder used to just be called Huntington's chorea. These are sort of dance-like, jerky, uncontrolled movements. The word chorea comes from a Greek word meaning dance, like think choreography. And then later on, it begins to affect the rest of your brain, and you have dementia as well. So here's a chart just showing the way this plays out. Years plotted along the x-axis here, and percent of normal function is plotted along the y-axis. So if you look at functional abilities, like your overall ability to do things that are needed for normal life, those drop off rapidly reaching kind of an advanced state later on in adulthood. Meanwhile, motor impairment, cognitive impairment, and chorea, those jerky, uncontrolled movements, all steadily increase. Usually the diagnosis on average, I guess, is made around 45 years of age. This is when the symptoms have gotten to a point where you can't really brush them off or ignore them. You really have to start saying, okay, there might actually be something wrong with me. I think with neurological conditions, oftentimes they can be they can be somewhat easy to brush off, because you don't really want to think about having a neurological condition, so you might ignore your symptoms or compensate for them. But after a certain point, it becomes unavoidable. So Huntington's is characterized by what I said, chorea, these jerky, uncontrolled movements, coming from the Greek word meaning dance. There's a range of cognitive and mood symptoms as well, many of which actually come before the motor symptoms. This is one of those things where typically it's the motor symptoms that bring people into their doctor for the first time. However, when you have made the diagnosis and you go and look retrospectively, very often you or your loved ones will say, oh, you know what? Before the motor stuff really got bad, he definitely had a mood change a few years earlier. Like he was crabbier or more impulsive or whatever. People's mood will often change, but it's a little tough to separate these neurological disease associated mood changes from the normal ups and downs of everyday life. Obviously, if you're in your mid 40s, you could be having a midlife crisis, or you could be going through some other kind of psychological challenge that has nothing to do with Huntington's. So people will look at the cognitive and mental symptoms on their own and perhaps not immediately leap to a brain destroying neurological condition. So Huntington's begins by affecting the basal ganglia, in particular the caudate and the putamen. These parts would be collectively part of the striatum, and also the globus pallidus. That would be more kind of classically in the basal ganglia. The symptoms, the motor symptoms, are primarily due to a loss of GABAergic medium spiny neurons in the striatum, so in the caudate and the putamen. But as the disease progresses, it is a neurodegenerative condition, so it's going to start to affect every part of the brain. It just happens to be the case that the basal ganglia are affected first. I remember when I taught, I guess it was 1203 this summer, I went into a whole big thing about the direct and indirect pathways of the basal ganglia and the different circuitry that's involved. We don't need to worry about that today. One of the funny things about studying the genetics and the genomics of mental illnesses is that oftentimes the neurobiology of it is kind of flattened out, where, the amount of genetics we understand about a given condition might potentially be enough to tell you what's going on in a certain cell in a certain part of the brain. But to get to a circuit level explanation at the level of sophistication that neurologists have when it comes to this condition, that's quite a stretch. So we can flatten all that out and just say this is a problem due to neurons dying in the striatum and elsewhere in the brain. And you can see that the neurons die elsewhere in the brain by comparing this coronal section of a Huntington's brain to somebody who's the same age but does not have Huntington's. These are obviously post-mortem samples, but you can see the Huntington's brain is all shriveled up. These sulci between the gyri of the cortex are all big. This is due to a shrinkage of the brain material itself, the gray matter and the white matter. The striatum, and the basal ganglia, which normally lives here, it looks kind of like an ice cream cone, or it looks like kind of the shape of a piece of candy corn, like this sort of like layered triangle kind of thing. But anyway, that's all shrunken down, and the whole brain just looks rather bad. The ventricles look big. Typically, when you lose neurons and brain material, the ventricles get bigger. So not great, folks. Not something you would want. A very, very nasty neurodegenerative condition. And what we will learn is that all of this is because of one single mutation, a trinucleotide repeat expansion in the Huntington gene, HTT. This is a coding mutation, so I think that's an important thing to bear in mind. The fragile X trinucleotide repeat is not a coding variation, so that's a big difference between the two, despite the fact that the actual type of mutation is kind of the same. This causes the translated protein to have extra glutamine residues, and this makes the protein behave differently. It causes it to aggregate in neurons, and this aggregation of the defective proteins is cytotoxic. It's bad for the cell. It causes the cell to die. So that's sort of the way it works. Now let's go and take a little bit of a history lesson, or kind of walk through the progression of how this disorder was characterized. And this is one of the great privileges we have in the two trinucleotide repeat disorders, because they're monogenic conditions with very defined, well understood mutations. We can actually go through the whole history of the discovery process and see how they got from noticing the disease exists in the first place all the way to developing a genetic tool to diagnose whether somebody has it or not and understanding exactly how it works. That's a privilege we don't have for things like schizophrenia or depression or anxiety or personality or any of these other more complicated things. The hope, I guess, at some stage in our history was that everything would work this way and you'd be able to find mutations that could explain everything. In reality, most of the conditions we know about in the mental health world don't work this way, but fortunately a few of them do, and we can learn from that. So Huntington's is a monogenic disorder, and let's just take a look for a moment at this pedigree. So the black shapes are people who are probans, or people who have Huntington's. Hollow shapes are people who don't have it. Slash through the shape means the person is deceased. And this was first noticed by a fella named George Huntington, who died in 1916, so midway through the First World War. He named the disorder. It had actually been described by earlier researchers, but he was the fella that named it. And the way that he discovered it and the way that people understood this for quite a while was just by looking at these pedigrees. And you can see that if your parent happens to have this, your odds are quite good of you yourself having it. Somewhere around 50-50. It doesn't exactly look like 50-50, or, the odds seem kind of unfair here. But strictly speaking, it's about 50-50. So here you can see an example where these parents had 5 kids, or no, how many? 1, 2, 3, 4, 5, 6 kids, and about wait, am I counting that right? 1, 2, 3, 4, 5, 6. Yeah, they had 6 kids, and 4 of them had the condition. So that's not quite 50-50, but it's sort of approximately so. Anyway, the pattern that we're talking about here is autosomal dominant. So you can kind of identify this whenever the odds of a child inheriting their parent's trait is about 50-50, regardless of what sex the parent is, then that's the pattern that's a pattern consistent with autosomal dominance. If the pattern had to travel on one of the sex chromosomes, you would see it could only be inherited from a parent of one or the other sex. So that would be a sex chromosome or an X dominant or a sex-linked trait. But this is an autosomal dominant condition, and it runs in families very clearly. And I guess I should point out the fact that it runs in families is kind of counterintuitive, I suppose, because it's a very severe condition. It's the kind of condition where if it happened maybe, if the onset was 10 or 20 years earlier in somebody's life, in all likelihood the condition wouldn't exist in our world because people who had it wouldn't successfully reproduce and pass those genes on. Genes that are deeply deeply pathological and actually prevent the person from living enough of a normal life to reproduce, those genes do not tend to stick around in the gene pool. They naturally are selected out. That's part of the reason why most of the conditions we have aren't that bad. Huntington's is kind of a unique case because it it appears later in life, oftentimes after the person has already gone to the trouble of reproducing. So the person may have passed their genes along to their kids without even realizing that they themselves had the condition. So this allows it to kind of slip under the goalie and make it through the normal process of natural selection, despite being a very bad and deeply pathological state of affairs. Anyway, so back in 1983. It had been known, at least, that this Huntington's condition or Huntington's disease was a autosomal dominant trait, and based on various aspects of the pedigree and the available genetic evidence at the time, it was pretty obvious that it was just one gene located somewhere in a specific place somewhere in the genome. So, in '83, Gusella and company mapped the location of the gene, whatever the gene was, to chromosome 4. Now, they could using the technology they had, they knew that they could locate this gene somewhere on the chromosome. They could locate, its approximate location, what band of the chromosome, and like what arm and what band of the chromosome it happened to be on. But very little beyond that. However, they did have probes, oligonucleotide probes, that could detect the mutation, and therefore they argued that you might be able to develop a molecular test to determine whether somebody was a carrier or not. The next thing that happened was 10 years later, or the next major development was 10 years later. The Huntington's Disease Collaborative Research Group, led by a whole bunch of people working in this field, was able to map the gene out and determined that the mutation which causes Huntington's is located in a novel gene. So this might seem a bit trivial, I guess, to think of now. You can go to the UCSC genome browser and you can type in any coordinate you like and see exactly what genes happen to be there. But this is not how things were in those days. Some genes were known about by way of linkage studies and by way of very tedious efforts by examining pedigrees and patterns of linkage and so forth. But most genes, nobody had a clue where they were located. I mean, people didn't even know how many genes there were. So to discover that the mutation that causes a disease is located in a gene, and more importantly, a novel gene that nobody had ever characterized before, that is a fairly major advance for the early 90s. And what's more important, the authors identified a region of this gene containing a trinucleotide repeat element. So a trinucleotide repeat is simply it's a stretch of the genome where you have the same three nucleotides being repeated multiple times. In this case, the nucleotides are CAG. So you just have CAG repeated many, many, many times. In normal people, or in normal individuals who are healthy, the number of repeats you get would be about 21. It's near the beginning of the gene, near the transcription start site of this gene. So you get about 21 repeats in the typical healthy person. However, this repeat area was massively expanded in Huntington's patients. You'd see as many as 100 repeats. So over here is the sequencing gel from the paper they published on this topic, where they're trying to count up the number of repeats. So this is from Sanger sequencing. I guess you can kind of, it's maybe a little helpful to look at this because it might be the first time you've ever seen how Sanger sequencing is read out. But essentially, you can go back to the sequencing lecture to learn about how this is done, but they're using terminator nucleotides A, G, C, and T. And so wherever there's a band, that is a location along this stretch of repeats where the reaction happened to have been terminated by the addition of an A. So wherever you see a band on this gel, at that position, there is an A in the sequence. So all the way down here at the end are the shortest fragments, and all the way up here are the longest fragments. That has more to do with the size of the overall piece you're amplifying, not so much the nature of the genome. But anyway, what you see is that you have an A, and then you have a G and a C. And these bands come up in kind of a stair-step fashion because one nucleotide always follows the next. And this very stereotyped repetitive pattern is exactly characteristic of the CAG repeat. If it had been a regular part of the genome, these bands would be in all kinds of different places. You'd see T's. Here you see no T's, so there's no T's in this repetitive area. That part of the gel is empty. So you can see that this repeat length has a certain sequence to it, and it also has a length that's a good deal longer than the average person. So here's the same region in a normal person. They've got 21 repeats. Here's the region in a person with Huntington's. They have 48 repeats. So this is, just, I guess, an example of how Sanger sequencing was used in real life back in the early 90s. This was a technique. Now, I mean, to do this is very old-fashioned, and it wouldn't really be something anybody would ever need to do at this stage of the game. But back in the olden days, it was quite common. So the Huntington's gene, the HTT gene, it's the gene that causes Huntington's, more or less, and it contains a CAG repeat in the first exon. So the first exon is the first coding region of the gene. And it just so happens to be that CAG is a codon. A codon is a sequence of 3 nucleotides that specifies a certain amino acid in the protein coding language. So if a ribosome reads CAG, it will add the amino acid glutamine. So the more CAGs you have, the more times glutamine gets added into the to the growing protein. This yields what's called a polyglutamine segment, also called polyQ, not polycule, polyQ. This is the Q is just sort of the shorthand code for the amino acid glutamine, so you can call it a polyQ segment if you like. But anyways, you're adding a whole bunch of these glutamines that aren't normally supposed to be there. And what, if we know anything about proteins, it's that your molecular biology, your cells, are very particular about having the right sequence of protein. Think about the impact that a coding mutation at one nucleotide, a SNP, can have on a protein. It can completely change the function of that protein in a fairly major way. So imagine just taking the normal sequence of the protein and throwing in a whole bunch of extra glutamines that aren't even really supposed to be there. That can pretty severely disturb the way that protein is supposed to work. Now, these repeat lengths show variation between individuals. So this is an area of the genome that is quite variable, as is, I should point out, and I mentioned this several lectures ago, I suppose, repeat length polymorphism is a big part of human genetic variation. This was what was used in linkage studies with those microsatellite markers. So it's normal for repeats to vary between people. They're quite variable because repeats can expand and contract very easily. It's a type of mutation that happens quite commonly. So there's normally variation in the length of these CAG repeats in the first exon of the HTT gene. Fewer than 27 repeats and you're fine. If you have between 27 and 35 repeats, you have an elevated risk of passing Huntington's onto your offspring, but you yourself will be okay. See, the longer the repeat is, the easier it is for a repeat length expansion to happen. And also, the easier it is for the repeat length expansion to push that number of repeats into the dangerous zone. So if you have sort of an elevated number of repeats, you yourself will be fine, but your child might be at a higher risk if the repeats expand any more beyond that level. 36 to 39 repeats may or may not cause Huntington's in you, but is very likely, or more than 50% likely, to cause Huntington's in your offspring. And then greater than 40 repeats will cause Huntington's in you, the individual, and of course it will also cause it in the offspring. So you can actually genotype this. This is PCR followed by gel electrophoresis, and you can genotype the repeat length. Typically when you do electrophoresis and you're watching DNA migrate down the gel under the influence of an electric current, the shorter bits of DNA migrate more quickly. So if you run the gel, say for a half hour, the shortest pieces of DNA make it the furthest in that length of time and end up near the bottom of the gel. The longer pieces migrate more slowly through the agarose gel, and thus they stick closer to the top. So when you run this, you could see that somebody's repeat length down here would be shorter, and somebody's up here would be longer, and that's one way that you can diagnose this ahead of time. Because these repeats are present in every cell of the body, not just the brain. So you can actually go and take DNA from somebody's blood or from somebody's buccal cells, like lining their cheek, and you can determine with a completely, with a 100% certainty, whether or not they will someday develop Huntington's, even if they haven't shown any symptoms yet. So you can do this, and if you have Huntington's in your family, you can go get this test done, and the test will tell you with absolute certainty whether or not you will develop Huntington's in your life, and with some level of certainty whether you risk passing it on to your child. Again, they simply count the number of repeats you have, and they look at these probabilities and say, oh, you've got, 37. You're probably okay yourself, but the odds are pretty good you're going to pass it on to your child. You might want to think about some choices to be made there, kind of thing. This is the work of genetic counselors, which is a growing and very interesting career choice, is interpreting these data and helping people understand the results of their tests and so forth. We'll talk a little bit about, by the way, the implications of this test on people's kind of subjective first-person experiences, because I think that's relatively important, and this is a great time to bring it up. So here's just kind of a survey of the pathogenesis of it. You have this HTT gene, the Huntington gene that's located on chromosome 4, and in the first exon you have this CAG repeat. Now, there's kind of two ways that this repeat can affect the function of the protein. The first is you could end up with the gene being transcribed in full, and you have this Huntington's mRNA that's translated, and you end up with this extra long protein that is by virtue of it being too long is cleaved, and you end up with Huntington fragments that cause all of the cytotoxic effects. It's also possible for your cells to only transcribe the first exon and translate that. And here again, you end up with these fragments that can aggregate in the cell and behave in a rather cytotoxic way. So we just got a question in the chat. You may address this later, but do higher numbers of repeats correlate with stronger symptoms of HD? Somewhat. We do get into this a little bit. The higher numbers of repeats modify the age of onset. So the more repeats you have, the earlier the disease will kick in. The overall severity is almost, I think, a little bit deterministic. Like once the disease progression starts going, it plays out along a fairly similar trajectory for everybody. But when it comes to like quality of life and like number of years of life lost, more repeats definitely equates to a more severe illness. So yes, the number of repeats does have an effect. Anyway, so again, this class isn't about the cell biology of Huntington's. It's suffice it to say that it produces a bad version of the Huntington protein. It's a coding variation, so it actually changes the sequence of the amino acids going into the protein. And this has all kinds of bad effects in the cell. And for whatever reason, we won't get into it, but for whatever reason, it happens to be the case that cells in the brain are the most sensitive to this. Consider the fact this is a genetic mutation. Theoretically, cells in your toenails and in your liver or in your earlobe or wherever else also could theoretically express this gene and have problems themselves. But for whatever reason, it's only expressed in the brain. What the protein does, I think, is still a little bit unknown. I think it's some kind of cytoskeletal thing, I think, or some kind of structural protein in the neurons, I believe. But that's one of the funny things about this is you can discover the gene and discover that it, if there's mutations, it's bad, but it's in a whole other ballgame to figure out what it is the gene is supposed to do in the first place. So I've told you, I guess on at least 2 occasions now, that trinucleotide repeats, whether they're microsatellites or whether, technically these are also microsatellites, but microsatellite repeats or trinucleotide repeats or any kind of repetitive region in the genome tends to be rather polymorphic. That is to say, it tends to vary quite a lot between individuals because repeat expansion is something that can happen fairly easily during a DNA synthesis. So this could happen during either meiosis or mitosis, although when you're talking about the heritability of Huntington's, you're usually thinking about meiosis. So it's not unusual, if you think about the way DNA is copied, it's not unusual for the DNA polymerase molecule to occasionally lose its place. Now think of like the DNA polymerase is essentially taking the template strand and it's coming along and it's adding base pairs onto it to create the daughter strand. So it's going along and it's copying the DNA, but every once in a while, for reasons that could be varied, the polymerase molecule will sometimes pop off and have to, and the whole process will be temporarily paused. While this is happening, the growing daughter strand is kind of flapping around. It hasn't yet annealed to the template strand. As it's flapping around, it's possible for the daughter strand to base pair or to anneal with the template strand in the wrong place. Base pairing just depends on the bases matching up, right? So if this daughter strand is flopping around because the DNA polymerase enzyme has fallen off, it could reanneal anywhere where the base pairs happen to line up, okay? Now, ordinarily, in a regular part of the genome, this isn't terribly likely because as you go down the genome, every part of it is unique enough that base pairing really can only happen in the exact right order. for a regular gene, there's not any kind of stereotyped or repetitive structure, so base pairing can only really happen in the right order. That means that if the DNA polymerase molecule or protein happens to pop off, things will kind of flop around and it'll probably just reanneal exactly the way it should and everything will be fine. However, in a repeat, the reannealing doesn't follow that rule because you're talking about an area where there is no unique sequence really. You just have the same 3 nucleotides repeated endlessly. So maybe the 2 strands will line up perfectly and the end of the daughter strand will reanneal to the template strand in a 1-to-1 ratio. But it's also possible that the daughter strand might anneal a little bit further up the template strand, like this, because it's repeats. So, CCG just has to bind to GGC. The base pairing just has to line up, and because they're trinucleotide repeats, it could line up kind of anywhere. So there's no requirement that the 2 strands reanneal in the same relative position. So if the daughter strand reanneals in the wrong place, you end up with this kind of like loop or this flap of extra DNA on the daughter strand. This then you have that, and then you have the DNA polymerase enzyme is going to jump back on. It falls off for a moment, but it'll jump back on where it thought it left off and continue copying. So what do we have here? Well, we have a perfect copy of the template strand, except on the daughter strand, you've got this little extra loop of extra repeat segments flopping around. The next time this is copied, it's going to stretch itself back out and it will be longer than the original template strand because this loop will stretch out, and now you've got a template strand that has several more repeats in it than you started with. So after a couple of rounds of cell division, the number of repeats can expand considerably just by this polymerase slippage. Does that make sense? I think I've described it a little bit like if you're reading a very long repetitive passage in a book or something like that, it's very easy to lose your place because every part of it kind of looks like every other part. So if you lose your focus for a moment, it's very tough to remember where you are. I gave the example, which I think is a kind of a bad example, but of listing out the digits of pi because it's very easy to lose your place there. It's kind of bad because pi is definitively not repetitive. It's the exact opposite. But it's the kind of thing where like you're just reading through the list and you lose your place and then you don't really know how to get back to the right spot. And while this happens, you can gain a number of extra repeats. So this is the way that repeat expansion seems to happen. There are a few other theories and viewpoints on it, but this is sort of the favored explanation. Now repeats can also shrink, but it's much less likely. It's much easier for them to expand than it is for them to repeat. And the more repeats you start with, the easier it is for this to happen because the longer the stretch is of repeats where the possibility of slipping off and having this loop form, the probability of that goes up. So Huntington's depends on what we would call stochastic repeat length expansion. Stochastic in this context is kind of synonymous with random. It means that it happens without any predictable or it happens for reasons that aren't easily predictable. It just seems to happen on a roughly random basis, although stochastic and random are not exactly the same thing. It's functionally kind of the same as being random. So the biggest factor in determining how many repeats you end up with is how many you start with, because the more repeats you start with, the more likely it is for when the polymerase slips for the repeat count to expand. The baseline number of repeats forms the starting point. So this is why, of course, people with greater than 27 repeats, but not enough to get the full disease, so people kind of in the risk category, are at higher risk of passing Huntington's on to their offspring. And it also happens to be the case that if you look on a population-wide level, people with higher numbers of CAG repeats in the overall gene pool of that population tend to also have higher prevalence of Huntington's. So here's a figure showing that. This is merely a scatter plot showing on the x-axis the mean number of CAG repeats in the kind of general population. And then on the y-axis, the prevalence of Huntington's disease plotted in cases per 100,000. And what you will see is that people of European descent tend to have the highest number of repeats in their kind of background baseline population. And they also tend to have the highest rate of Huntington's. People of African or East Asian descent have relatively fewer numbers of repeats, and they have a much lower prevalence of Huntington's. I mean, it almost looks like it's 0 here. It's not, but it's quite a bit less. So this is, as far as we can tell, endemic to all populations. It's possible for any person on Earth, theoretically, to have it. It's much more common among people of European ancestry. And this has to do with the number of baseline repeats that they happen to have. Now, when you think about this process of repeat expansion, normally we're worried about the heritability, because when it happens during meiosis, you can convert a potentially harmless Huntington's allele into a pathological one. This is that case of people who are kind of on the border of having like enough repeats that they themselves are not really at risk, but enough that their child might be at risk if the repeats expand any more than they already are. And this rate of repeat expansion is a function of baseline length. So here is a plot showing their showing the number of CAG repeats in an individual's body, plotted against the number of CAG repeats you see in their sperm. So what this means is like in the number of CAG repeats you have in your somatic cells, the constituent of CAG, that determines your personal risk of having Huntington's. If you have more than a certain number, you will have the condition. I can't remember what the cutoff is even off the top of my head, but it's somewhere around 40 or 50, right? Maybe I should just look that up so I don't embarrass myself. So if you have greater than 40, you'll get Huntington's. So the cutoff would be kind of right around here. These are the number of CAG repeats in your body determine your risk. However, the number of CAG repeats, if you're a male, the number of CAG repeats in your sperm dictate the risk of you passing it on to your offspring. And repeat expansion can happen during meiosis when these sperm cells are being produced. So theoretically, if you have a high number of repeats, the odds of the number of repeats being expanded in your sperm cells also goes up. And this is precisely what they find. So if you plot the number of CAG repeats in the body on the x-axis and the number of repeats in the sperm on the y-axis, you'll see there's kind of a positive trend. It's a little bit below the level of what you'd call a strong positive correlation, but there does seem to be some movement there, such that if you have like a high number of repeats in your body, the odds are very good that you'll have a high number of repeats in your sperm, potentially even more. You can see the error bars of this can shoot up quite high, especially when you get into this high range here, where if you have 50 repeats, you could be passing as many as 80 along to your offspring if they happen to be made from that particular sperm. So the rate of repeat expansion accelerates the higher the number of repeats in your own body happen to be. Now, as I said, the course of Huntington's is, I think, fairly similar depending, regardless of the number of repeats you happen to have. Once you tip across that threshold to having the disease, it'll play out according to kind of a set pattern. Although I think that there would be, I think if the more repeats you have, it probably would make it worse and more quick. But one of the bigger factors that people look at are age of onset. So age of onset I think is important because if you have the gene or if you have this mutation, you're guaranteed to get Huntington's. There's no cure for it yet available. That being the case, you would at least like to have as many quality years of life as you can before the disease appears. That seems reasonable. Pretty much all of medicine, all medical efforts in the world are really just concerned with trying to increase the number of quality years you have available to live. That's the aim of most cancer treatments, for example, is to try to give you more years to your life. So we're quite curious about modifiers of the age of onset, because if you're going to get Huntington's, we might as well make it happen as late as possible so you get as many good years as you can. So people have looked at this, and the biggest modifier of disease onset happens to be the number of repeats that you have. So here on the x-axis of this plot, we have the number of repeats that you have on your CAG, or the number of CAG repeats you have. And on the y-axis, we have the age at which the motor onset began. So how many years old was the person when they first started developing motor symptoms. And what you will see is that the higher number of repeats you have, the earlier the motor symptoms appear. So if you have exactly 40, which is sort of the cutoff for when you start to develop Huntington's, your age of onset might be as high as 60 on average, maybe even 90 potentially at that point. who knows? But if you have 60 or say 55 repeats, your age of onset now is going to be like kind of your mid-20s. So that's a huge delta in terms of age of onset. Now, it's important to note that there's a lot of residual variance. This function plot, I think this is a very predictive function. The most important thing to know is the number of repeats you have, but it's still quite variable. I mean, if you have 40 repeats, your age of onset could be as low as 30, or it could be as high as 90. There's quite a lot of variability around that line of best fit. So repeat length is not 100% predictive. There's a great deal of unexplained inter-individual variation. Now, what's interesting is that this inter-individual variation, or this residual variation in disease onset, itself is heritable, but it's heritable distinct from the CAG repeat. So people with, say a certain number of repeats running in their family, they might have that number of repeats, but also be early onset people or late onset people. And their tendency to be earlier onset or later onset is inherited separately from the number of repeats. So there are other genetic factors at play that regulate the age of onset. And that's kind of interesting because we aren't really going to be able to do very much about the repeat length. It's not really possible to go into all of the cells in your body and delete those extra repeats, at least not using our, currently available technology. But if there are genes that you have that modify the age of onset, perhaps we could study the biochemistry of what those genes do and then develop therapies around that basis. So let's look at that a little bit more closely, because I think this does hold some promise. But we're going to have to take a bit of a detour first. So this is our concept of somatic repeat length expansion. So far I've talked about repeats as something that can expand mostly during meiosis, something that makes them heritable and allows them to expand, causing a sub-threshold case to be amplified to a above-threshold case in offspring. And we've also learned that all of our cells contain identical copies of the genome. The cells in your hair follicles have the same DNA as the cells in your retinas, as the same DNA as the cells in your gums or whatever else. Like these all have the same genome. But actually, the reality is this is only approximately true. It's only mostly true that all of your cells have the same DNA. The reality of things is that small mutations can build up over numbers of cell divisions in response to DNA damage, or like UV light, for example, etc., or as a result of transposon expression. All kinds of things can actually happen to make the DNA change in individual cells in your body, such that especially the older you get, not all of your cells are going to have identical DNA. Little mutations will build up between cells. In fact, this principle is what causes cancer. At least one of the main reasons it causes cancer is that cells are perpetually having individual cell autonomous mutations, and every so often you get a mutation in a tumor suppressor gene that causes the cell to divide out of control and turn into a tumor. So this is so the fact that our cells contain identical DNA is only roughly true. It's basically true, but it's not completely true. The tendency for different cells in your body to have different genomes is called mosaicism, and we're not talking about the web browser from the 90s. We're talking about the principle of mosaicism in genetics. So the word mosaic, I should say, comes from a Greek word meaning muse. These tiled patterns where you make a picture out of little tiny colored tiles, that's called a mosaic. So what they're trying to evoke here is an idea that like you yourself are made up of many different types of cells with many different types of genotypes, or many different genotypes. And if you were to imagine the different genotypes as being different colors, then you would see that you are kind of like a mosaic. You're made up of many different pieces, like the way these artworks are. Kind of an evocative term. But this is a basic fact of the way our bodies work, is that we do have some level of mosaicism as mutations pile up in different parts of the body. This, by the way, just as a point of interest, when they're doing genetic tests, for example, the GTEx Atlas, which is looking at genotype and tissue expression, they do make a distinction between sun-exposed skin and non-sun-exposed skin, because skin that's exposed to the sun, like, say, the top of your head, if you're like me, is going to develop a number, a great many more mutations due to exposure to UV light than skin where the sun don't shine, which is going to be relatively pure and much closer to the way you were, much closer to the DNA you were actually born with. So this is quite an important aspect of things. I mean, this is why you're supposed to cover up when the sun is out, because that's, these kinds of mutations can lead to skin cancer, and mutations happening in other parts of your body can lead to other kinds of cancers. But anyway, I kind of digressing here. The point is that not all of your cells have the same DNA. And what this implies is that the repeats, these CAG repeats, can actually expand in your body separate from the process of reproduction and heritability. So it was discovered fairly early on, actually, that the repeat length varied between people, but it also varies between tissues within the same individual. So this is intra-individual variation, where if you look at different parts of somebody's body, you'll count different numbers of repeats. And actually what you sort of find is that this is also true between people. The number of repeats that you count is kind of the average, like what's the most common number of repeats in all of their cells. But if you look closely enough, you'll find some proportion of cells have more or less repeats than the overall average, meaning that within the same individual, there's some level of variability between cells. So this is a PCR analysis of repeat length, and this is coming from the same donor. So they're looking at the number of repeats in all of these different organs, and and what they're saying is, like, notice the extra bands here. There's sort of a central band that represents the average number of repeats, but there are these little kind of satellite bands representing areas where there's more or fewer repeats in that particular tissue. So on average, the individual would have 40 repeats because that's the darkest band, but there are occasionally cells where you'll get higher numbers of repeats amplifying as well. These bands have higher molecular weight because they're longer repeats. And if you look across the different tissues, it's especially striking that the bands above the bands on the lower end are the low molecular weight ones. I got that mixed up. What I was actually trying to say is that the bands up here on the higher end tend to have a higher molecular weight and thus have longer numbers of repeats. And if you look across the different brain or across the whole body, what you will see is that the brain, the caudate and the putamen in particular, tend to have more of these high molecular weight bands. So these higher numbers of repeats tend to be enriched in the caudate and the putamen, which, as I mentioned, are precisely the brain regions where Huntington's first shows up. So it seems like there might actually be something special happening in this part of the brain that causes the number of repeats to increase even above the ones, even over and above the ones you were born with. Interesting to think about. There's also kind of a weird exception where this doesn't seem to happen in the cerebellum. The cerebellum actually has fewer repeats than other parts of the brain. It seems strange that repeats might actually contract there, or at least not expand to nearly the same rate. Here's another example. So here's somebody with Huntington's at I don't think that actually means age 6, but anyway, they have Huntington's that comes on at a certain age. I guess 78 repeats, you'd have a fairly early onset. But anyway, most parts of their brain, they have approximately 78 repeats in this person. But for whatever reason, in their cerebellum, they actually have 65 repeats in the cerebellar vermis and the cerebellar cortex. The vermis is just a little kind of midline structure that looks a little bit like a worm on the cerebellum. Vermis means worm. But anyway, something very funny going on in the cerebellum. We're not going to get into this, but I would say that if you're a brain enthusiast, or if you're kind of interested in genomics and epigenomics, pay attention to the cerebellum, because there's very strange things happening in that part of the brain. I've noticed them too in some of my own stuff. Very odd, weird things happening in the cerebellum in both the epigenetic and genetic world. And by the way, the cerebellum also has half the neurons in your entire brain are located in your cerebellum. And it's this weird structure that looks very strange and does things that people aren't even, anyway, like it's very funny, very strange things happening in the cerebellum. Anyway, the point I'm trying to make anyway is that you're born with a certain number of repeats, whatever your parents gave you, but then the individual cells in your body can expand this further as you develop, such that places like the caudate and the putamen, the parts of the brain that are especially prone to Huntington's, can actually have more repeats even than the number you were born with. And of course, the number of repeats dictates the age of onset. So you can have this variability where, maybe some parts of your body have the number of repeats you were born with, but other parts, like your brain, can have even more repeats than that, and that can make you have an earlier onset despite the genotyping not being able to tell, because you have to be able to genotype somebody's brain, and that's not easy to do. So here is the rate of somatic repeat length expansion, and it's we're looking at it in the context of young versus old onset. So they're taking tissue from the I'm not sure actually what organ this is, but it's tissue from some part of the body after the person's already been born and developed, and you have an age of onset. So over here, the age of onset for the first figure is 25 years old. The other person down here is 30 years old. Here in the older onset group, the age of onset is 41 and 72 respectively. And then here you're looking at the number of, or the frequency with which you see a given number of repeat expansions. So 0 means that in those cells, the repeats didn't expand at all. In 1 means the repeats expanded more than once, greater than 5, greater than 10, greater than 15, and so on. And less than 1 means that the repeats actually shrunk down a little bit. So what we're showing here basically is that if you happen to have a young onset, there's a lot more instances where the number of repeats expanded considerably, as many as 25 or 30 times. And this is also true for this other person that they looked at. But if you were an older onset person, the odds are pretty good that the repeats barely expanded at all. So the difference in these plots is really in this kind of pink area, where young people with young onset of Huntington's with the same number of repeats as a starting point, 41 or 47, the age of onset seemed to be mostly a function of how many times the repeats expanded. Given the same number of baseline repeats, it's the rate of repeat expansion in your body that determines the age of onset, or that seems to play a big role in the age of onset. This is okay. So this is brain tissue. Sorry, I didn't read this on my own slide. So they're looking at repeat expansion in brain tissue. So people with young onset, this person here started with 47 repeats. This person over here had old onset. They also started with 41 or 47 repeats. The young onset person had much more repeat expansion in their brain tissue, sometimes as many as 30 times above the normal baseline level, whereas the person with older onset had very little repeat expansion. Most of the time, the repeat only expanded by 1 unit. So that's the kind of point they're making here, is that there is this effect of how many repeats you start with. That has a big impact on how early your onset is. But then the next thing that has a big impact is how much the repeats have expanded during your life in your individual cells. So it's not just what you're born with, it's also how your body, how your body's able to handle that DNA and prevent it from expanding. Okay, so that's my point, is that repeat length expansion in your own body can also affect your age of onset over and above the effect of how long the repeats are that you were born with. So what's all this about? Well, modifiers of age of onset can be studied genetically using our favorite method, the GWAS. Same idea. You just take a whole bunch of people who have Huntington's, you genotype them, and then you do a study where you plot, or you have age of onset as a continuous variable, and then you just look for genetic variants that are correlated with it. And it's the same kind of thing. You've got your Manhattan plot here with your genome-wide p-value cut off here. Any points above this line are SNPs that are significantly associated with age of onset. And what we will see is that there is a cluster of SNPs on chromosome 5 that is strongly associated with your age of onset. Again, over and above the number of repeats you start with. Obviously, if you have 1,000 repeats, you're going to have a very early age of onset. But within the number of repeats you have, there's a whole lot of range. So this is a study of the genetics that mediates that inter-individual variation in age of onset. And so here's this region zoomed in, and so you can see the SNPs that come up above the p-value threshold are all in strong linkage with each other. That's what the red color means. They're all in strong LD. And they're located over this cluster of genes. So this is the genome browser view. So we've got MTRNR2L2, we've got MSH3, and we've got DHFR. So those are sort of the 3 genes where these SNPs are located. And you can do the simple thing of just asking, what are those genes and what do they do? Right? I mean, that's a pretty basic question. What, the authors are lucky in a way. They only found one place where there's any significance. So all of the, their only hope in explaining anything lies within this area. And odds are pretty good that it involves one of these 3 genes. So let's take a look at what those genes do and think about what that could mean. So this is Moss et al. And I'll just cut to the chase. They found a coding variant in the gene MSH3. That's this one right here. They found a coding variant there, as well as others in the same kind of neighborhood, that were significant modifiers of the age of onset. They also used data from GTEx, which is the genotype tissue expression database. And they showed that the SNPs that influence disease progression also act as eQTLs, meaning that they affect the expression of the associated genes. So these are SNPs that have an effect on both the coding and the expression of nearby genes. So what does MSH3 do? Well, it plays a role in DNA repair. It codes for part of the DNA mismatch repair system. So this is the kind of the very exact thing that can mediate repeat expansion. When the repeat is partly unwound, the unwound DNA is normally bound to by a dimer or a complex of MSH2 and MSH3. It will cut out the offending section and fix it, and this typically leads to an expansion of the CAG repeat. Oftentimes, even when you try to repair repetitive regions, they tend to get longer, just because, again, the enzymes all lose their place. Without getting into it in too much detail, that's what's happening. Is this part of the DNA repair system actually can cause more repeat expansion? So the authors are arguing that these mutations to MSH3, I don't know why I have so much trouble saying that. I think I'm trying to say MC4R, another gene that I study, or MC3R, MSH3. Anyway, they're arguing that mutations to this gene affect its ability to repair this unwound DNA, and this can actually increase the rate of somatic repeat expansion and thus cause earlier disease onset. So I think this is a terrifically it's a terrific story, I guess, in some ways, because they're doing a GWAS study. They could have found genes anywhere. They were looking across the whole genome. They could have found genes in some random place that had nothing to do with the DNA, but it just so happened that the genes they found are all located in a DNA repair system and gene, and DNA repair is precisely the kind of thing that could lead to somatic repeat expansion. So the argument is this. Repeat length expansion in the adult brain, particularly the striatum, can lead to earlier disease onset, because it's not so much, it's the repeats you're born with are, important, but what really matters is how many repeats are actually in your brain when you're walking around in the real world. The more repeats you have in your brain, that's what really matters. You could have a totally normal number of repeats in your arm or some kind of other somatic tissue, but if you have a huge number of repeats in your brain, you will get Huntington's. So they're simply arguing this. These MSH enzymes can be are mutated variations in these are correlated with age of onset. These repair the DNA during cases where the DNA is partly unraveled or partly unwound or something else is going wrong with it. And this repair process going wrong can cause expansion of this repeat tract. And the more times this happens, the bigger the number of repeats gets. And accordingly, the earlier the age of onset. So the repeat length you inherit from your parents only roughly predicts your likelihood of, roughly predicts your age of onset. You might get 40 repeats from your parents, but if inside of your striatum it's expanded to 55 repeats, your age of onset is going to be very, very early, even though according to the analysis of the DNA in your arm or your blood or whatever, you have a fairly low number of repeats. So the starting point matters, but what really actually makes the biggest difference is how many repeats you have in your brain. That has the biggest effect on your age of onset. So that's all I really wanted to talk about with the brain. I think it's a wonderful kind of it's a little bit nice when things all work out this way. They don't always work out so nicely, where you have a single monogenic condition that unequivocally leads to a given outcome. And it's found in the early 90s and understood pretty thoroughly by the late 90s or by the early 2000s. And then you find that the thing that causes a lot of inter-individual variation in disease onset happens to be an enzyme that perfectly fits with your understanding of the molecular biology of that trait. It's just great when that kind of thing happens, although there's a lot more to be learned. But I guess what you could say is that if you could find a drug to target this MSH3 pathway, or somehow affect the DNA repair machinery in such a way as to prevent repeat expansion, you might be able to hold off the disease a fair bit of time, particularly if somebody happens to have the kind of mutations around MSH3 that can lead to the disease coming on earlier. So that's all I really wanted to tell you about that. I don't think we need to get into the cell biology of how Huntington's actually works. That's something you would be able to learn in a different class. This is just some genetic information about Huntington's that will hopefully help you if that's a topic you're interested in. So I just want to finish off the class by getting into some questions about ethics. And I don't think, we're not trying to do a social studies class, but we are learning about genetics, and we're learning a fair bit. When you're finished your time at Carleton, you will know more about the genetics you'll know more about genetics in general and more about the genetics of mental illness than almost anybody else you'll ever meet, because most people don't go through this exact program that you're going through. And most people, maybe you'll only hang around with neuroscientists. That's one thing, but more than likely you'll enter the regular world and you'll be dealing with normal people who have normal interests, not these weird ones. So you're going to be a carrier of knowledge, whether you like it or not. You will be the people that, you will be the person that your family and friends will come and ask when they have a question about genetics or about these kinds of things. So it is kind of incumbent on you to know enough about the scientific side of things and also the ethical or kind of philosophical side of things to be able to approach these topics with the right level of nuance and grace. That's my opinion anyway. I think, it's important to consider things from all different angles. Ultimately, genetic technology is going to determine the way the future plays out for humanity. And now that you know more about it than most people, or now that you are, by the time you know more about this than most people, you're going to be in charge of somehow steering that kind of discussion. So I just want to sometimes occasionally bring up these kind of more soft issues just to give you some food for thought and make you consider the things that are probably important. So Huntington's manifests later in life. However, it's very deterministic. If you have more than a certain number of repeats, you're basically guaranteed to get it. Plus, you would likely know by looking at your parents whether it runs in your family. Each child in a family where Huntington's is present, or where the parent has Huntington's, each child has a 50-50 chance. So you know just at a baseline that you're basically a coin flip away from having the disease and having your life massively shortened and curtailed. But you can know with even more certainty by getting genetic testing done. It's very simple to get a test for CAG repeat expansion. You can have it ordered by your doctor. You'll get the choice to do it. And if you do the test, you will know with complete certainty whether or not you will develop Huntington's. This is one of the very few conditions where you can have such a perfect level of prediction. Of course, they can't tell the precise age of onset, but if they know the number of repeats you have, they'll give you a ballpark. They'll say, oh, you've got 40 repeats, you might make it into your late 40s. You've got 70 repeats, you're probably going to start getting symptoms in your early 20s, that kind of thing. We are aware that there's modifiers of age of onset, but they'll give you a ballpark. So this leaves people in a rather difficult position. Should you get tested? If you know you have a 50-50 chance, do you want to get tested, or do you want to not get tested and just try to live life to its fullest? So for those of you playing along at home, I would ask you just to reflect, what would you do if you were in this scenario? What would you think is the best choice? Would you want to know, or would you rather live without knowing? I don't know. It's everyone's. It's anyone's guess. And obviously, I don't. This isn't something that has affected me personally, so I can't. It's almost hard to imagine what I would do. I think that's probably hard for anybody who hasn't faced severe disease in their life or severe disability or anything along those lines. Best thing we can really do, I think, is just listen to individuals who have had it done and learn from what they have to say. So we have, there have been a number of papers looking into this, but I picked a few choice quotes out from McLeod et al. In 2014. And this is a study based in the UK, I believe, where they simply interviewed people who had Huntington's running in their family and asked them to reflect on getting the test and, if necessary, what their results were. So I want to just go through and just kind of read these. The overall feeling among the participants, this was just a survey, essentially, sort of a qualitative, like a structured qualitative psychological study. Most of them felt that it was a pretty easy decision to make. They didn't think it was too hard to decide to get tested, but adjusting to the results was a challenge. So here's a male, 19 years old, who tested positive for HD. What that means is that this individual is going to begin developing motor symptoms later in life. Maybe they would have 10, 15, 20 years before things really start to pop off, but they are going to start developing symptoms, and they will be severely impaired by the time they're in their mid to late adulthood. So this person writes, at the time I felt quite adult. You always do, don't you? You know the I know it all kind of thing. I'm glad I didn't have it when I was 15. So imagine that, the difference between somebody when they're 15 and 19. By the time you get, that's a huge difference in terms of your overall maturity and life outlook. But I think both 15 and 19-year-olds are very, very young, basically children in a way. And to have that type of knowledge when you're so young is really something I can't even conceive. Normally, if you're one of the fortunate people out there, you can look forward to a very long life without any, you don't predict any severely bad things to happen. This person is 18, also tested positive. She writes, I knew I had to because I knew I'd spend my whole life wondering, will I? There was never any second doubt that I was not going to get tested. I don't know. I think because I thought about Huntington's disease so much that I thought I just was going to do it anyway. So this person couldn't get it out of their mind. I think it would be hard to do that if you knew you had a 50-50 chance. It would be very tough to not think about it all the time. Another male tested positive. They write, I've got good faith in medical science. It's only going to get better the more research goes on. So a bit of optimism there. That's good to see. Here's a female who tested negative. When I got told I didn't have it, Huntington's, there were tears of happiness, but then for my sister as well. I felt really bad for her then. I've had times when I've really thought about it and I've got upset. The fact that my sister has got it and I haven't, and I always think to myself, what does she feel about me now because I've got it, or I haven't got it and she has. So here's a girl whose sibling tested positive and she tested negative. So how would that make you feel? Here's another female who tests negative. I convince myself I got it because I'm so like my mom. I look like her, I act like her. I could see myself in her even at that age. I think when you're young, you do think like that. So she also tested negative. I guess that's kind of an example of how linkage works. You inherit bits of traits from your parents, but not everything wholeheartedly or wholesale. You might get your mom's face and your mom's laugh, but you may not get her Huntington's gene. So that's something to consider. And then finally, a female of 21 who tested positive. There are negative things, but focus on the positive, which helps a lot. You might not get it young. I was worried that I was going to get it young like my mom did, but I might not. So here's somebody who tested positive, and by the sounds of her mom's situation, has a fairly high number of repeats, meaning that the onset could be rather young. She, however, is hopeful that she won't. Obviously, there's a lot of inter-individual variation in rate of onset, but it's definitely a tough call because odds are pretty good that she would get it at a similar age as her mom got it, given that she inherited the repeat length from her mom. So that's just a few qualitative remarks on it. I think it's, whenever you can, it's nice to think about these issues and try to gain some insight from them. Interestingly enough, parents oftentimes don't want their child to get the test. So for Huntington's especially, because there's nothing that can really be done. And I would imagine from the point of view of parents, there might be a bit of guilt there as like, what did I pass on? What have I done to my child? I've cursed them with this horrible disease. Parents may not want their child to find that out. Now, interestingly enough, the BRCA1 gene, which gives you kind of your risk of breast cancer, parents do typically want their child to get that test because there are things you can do if you know you have that mutation or if you know you have that gene to mitigate the risk of breast cancer and ovarian cancer. So I think it has to do with like how mitigatable it is. There's ways you can deal with some diseases, but there's not really a whole lot you can do about Huntington's at the moment. Anyway, that's all I really have for you today, folks. We're finishing up a bit early, which is always, which I'm sure you're just totally fine with. But yeah, this is Huntington's, and we're going to get into Fragile X next week on Monday, and it's going to be kind of following a similar kind of thing where you've got a trinucleotide repeat that is heritable and has a tendency to expand. We'll learn all about that going forward. So thanks to everyone for listening today. I hope you all learned something. Remember to smash that like button and share and subscribe and all the rest of it. Actually, don't share. I don't think you're supposed to share. Well, you probably can share. I don't think it really matters. Why not, right? The videos are unlisted, but I don't think there's any harm in it. But certainly do all the YouTube stuff if you feel like it. And I'll be holding office hours tomorrow at the usual time in the usual place. You can check your announcements for that. Don't forget about the assignment, and we'll see you all next time. We'll see you all down the road. Thanks for listening.